Geoff for inviting me to be able to participate in this session. I am new to webinar, so I don't know all the tricks that we have explained to us from Matthew, but as much as possible I'll try to do the best that I can to tell you a little story about my sort of passageway through teaching histology traditionally, and then moving that traditional teaching to suit the more self-directed learning styles that students now have, given the new technologies that are now available. First of all, let me explain to you what histology is, because I'm aware that many of you may not teach histology. Histology is the study of human cells, tissues and organs, or in fact any cell, tissue and organ. It could be the human body, it could be plant biology or whatever. It's an important subject for students to learn in any biomedical science course. Doctors, dentists, etc. They need to understand how cells and tissues and organs work in the body so that they understand pathologies, how to recognise tissues and organs that have gone wrong, that have had pathologies. So it's an essential component in all these courses. It's presented in a number of ways, and on this first slide you can see here, the top image of the microscope was the way in which histology has been taught ever since it was really a university topic. Students traditionally sat at a microscope, they looked at slides, sections of body organs and tissues, and they had to look down that microscope and try and identify all the characteristics of that particular tissue or organ, either following a laboratory manual or first of all listening to a teacher explain to them all the sorts of components in a pre-lab session. They also had lectures as well. Now, traditionally, it was taught as a purely structural course. And if you look at the image on the top right-hand side, students found that quite difficult to understand, in fact, quite boring. They didn't like histology because it was difficult. Now, there has been a transition because we have the advent of the computer, whereby students, as you see on the bottom right-hand corner, they can become more interactive with histology learning. And coupled with that has been the transition of teaching histology as a functional course, combining structure and function, which has made histology far more interesting to students now, and they actually have fun learning histology given all the new resources that are available to them. Histology is very expensive to teach. You have to give lectures, naturally. They need to have a textbook, an expensive textbook. They need to have a laboratory manual to guide them through the use of the microscope and looking at all the different sections of the body, sometimes 200 sections of various organs and tissues. They need to be able to use microscopes to look at those slides. They need to have slide collections, which means universities have to buy microscopes and they have to create slide collections for students to look at. These slides are prepared by technical staff within each school. Students use them and often break them, so slides have to be replaced. So it's an enormously extensive workload for schools that teach histology. And coupled with that has been the rise in student numbers. I had about 50 students 10 years ago studying histology in one of my courses. Now I have 375. So that means a university has to increase the numbers of microscopes and the numbers of slide collections. Very expensive. So that means duplicating, triplicating laboratory practical classes, which is enormously expensive in terms of staff teaching hours. And our academic staff and support staff are ageing. There is a new breed of teaching histology that requires younger staff becoming more adept with using all the new technologies. The reason why I'm telling you all this is because I want to give you some background in the histology courses so that I can then tell you about assessment. When histology is transferred to an online or a computer-based learning resource, there are a number of advantages listed in the slide that completely change the way in which histology can be taught, the effectiveness it can be taught, cost savings it creates, and more fun for the students. And I don't want to go through all the topics listed here. You can read through those on your own. But a couple of things very importantly I want to mention, and that is that students can go and look at these resources any time they like during the week, during the day. Whereas normally they would have been confined to going into a histology classroom to access a slide box and microscopes within a very confined timetable. They certainly couldn't do it outside normal hours. Let me draw your attention to this web address, which I don't want to go through now. Unfortunately, a lot of my web resources is very dynamic, and rather than try and test it on this system, I thought I'd just show you still images of the system. But I refer you to this website that you can go to at any time. If you email me, I can give you an enrolment key to look at how the courses are run. There are videos, or there will be videos in the next day or so explaining to you some of the components of this interactive resource. So I'd recommend you look at this course and see the interactivity and the new generation of histology teaching using technology. So what I did was I transferred all the learning online, or virtually online. And because of that, it enabled me to develop a more innovative means of assessing the students. When students register on this website that I previously showed you the URL for, there are two entry points on the website. First of all, they can go into an online interactive atlas, which is the first thing I want to very briefly explain to you before I get into the assessment. Secondly, that interactive atlas has over now got 7,000 images within it, and the interactivity allows students to interact with the images, to get labelling, explanations, etc. They can also use a computerised microscope and view a lot of their sections using virtual microscopy. They no longer need to go into a laboratory lab anymore and use a microscope. They can do it at home, in Bali, if they're away, or anywhere they like if they're ill. It's freed up laboratory space in our department and freed up cost of running a histology course. And the students keep saying it's more fun that way. Secondly, they can enter another link which gives them all the information they need. They can access lecture notes, audio-visual recordings of lectures, PowerPoint presentations, everything, including the assessment packages. So virtually, what was once a practically orientated course requiring laboratory time, laboratory resources, has now been transferred completely online. When you go into the atlas, this is what the students see. On the left-hand side, you see a list of contents, which is a normal histology course. There's about 25 topics there, which go through a whole year, academic year. Sometimes students would study the basic tissues in the first part of the year, and then the organ systems in the second part of the year. And as you see on the table of contents, there are these quizzes embedded in the table of contents. To briefly show you what the content is in this atlas, if a student clicked on a topic like skin, what would happen is that immediately a frame would light up in the centre which provides normal descriptions of skin that you would normally read in a very expensive histology textbook. Now you can access it online through this resource. You don't have to buy an extensive resource or textbook. Students would read through this material and it would give them the background to then do the laboratory practical or interactivity online and then complete the assessments. If they click on the little plus sign next to skin, they get taken to two things. One is the tree opens up. You can see in the table of contents on the left-hand side that the skin module or topic has now been opened up to a number of different topics, items of histology of the skin that students need to understand and understand very well. If the student clicks on any one of these items, for instance, if they click on the Meissner's corpuscle link on the table of contents, an image is loaded with some text on the right-hand frame. And that image is a very high resolution image of a Meissner's corpuscle. It's a very important receptor in the skin that helps us detect touch. Students can interact with the image by clicking on the hyperlinked blue labels you see in the text, and it will label various components on the image. So they can learn the histological characteristics of each of these histological items by interactivity between text and the image. And again, they can do this at home. But there is this tutor link at the bottom. And let me explain what that's for. If they go to that tutor link, it loads up their exact slide in their slide box. This happens to be a slide of their blood smear. And down the bottom you notice there is a microscope lens, a little microscope with 3 lenses on it. If students put their mouse above the lens, a bomb site, what I call a bomb site, comes up on the image. And that bomb site tells the student where to go when examining a slide of blood smear to find the particular cell types that they may be looking for. And if they keep clicking on that lens, it takes them up to higher magnifications and shows them the cell in detail. So it simulates using the microscope. And traditionally, they could do this at home and then come into a lab and very quickly go through the lab in perhaps a half an hour or 20 minutes, compared to the 3 hours they may have taken before they had access to this sort of resource. Here again is the bomb site showing them where to look in the slide to see a particular feature. Now, can I just pause for a moment and ask Matthew, is my voice coming across clearly? Can everyone hear me okay?
Yes, it does crackle a little bit sometimes, but by and large it seems good. Thank you.
Okay, thank you. I'm sorry about the crackling. I am all the way over in Perth, you know. Let's get back to this slide of the Meissner's corpuscle. This year, something else has changed, and that is that the student can now not only get stilled images of their slide, but they can also access the slide using virtual microscopy. Here is a web scope image of a histological section of skin. Now, if you could access this web scope section, and if you want to know details of how to do this yourself, certainly email me, but students can now go to a database or a slide collection using their computer, and they can examine the histological slides in front of them using the computer and using web scope. They can zone in on this section of skin at different magnifications and find details of the Meissner's corpuscle or layers of the skin. They can study microscopy now online using computers. They no longer need microscopes. And again, as I kept saying before, this has been an enormous cost saving for our school, particularly when we've only got 50 microscopes to suit the 50 students we had a few years ago, compared to now 375. So they access their learning completely online. Let's talk about assessment, but first of all, I want to show you this other content learning entry area. They can go into this and they access, again, a table of contents, and then in that table of contents, each topic has an enormous number of links that provide them all the learning resources they need for the study of each topic. And again, if you email me and access the Meyer's Histology on the Moodle site, you can see for yourself all the sorts of resources that are available. So what about assessment? And I'm sorry if I've taken too long to get into this, but I think it's important to explain how histology was taught and is taught now before I do that. Well, when you assess students in histology, you've got to think of what do they really need to know? They need to know how to identify all the organs and tissues of the body. They need to be able to identify the different cells in each of those organs and tissues, and they need to know about how all those cells and tissues and organs function properly. So a lot of the assessment is not sitting down in a lecture hall or a gymnasium doing a lot of written answers to a test. There is some written assessment, but the majority of the assessment has been students looking down at stations of microscopes and going from one station to another to identify organs and tissues. And that's a very time-consuming exam to create and set up. Typically, for an exam, traditionally, you would set up a microscope with a slide on the stage. From an exam collection you may have, there may be 40 slides set up. You'd have 40 students come in. They'd spend a minute or 2 minutes at each station and go from one station to the next and identify what tissue or organ is under the microscope, because they need to know that if they're going to do pathology. Also, they might go to a microscope, and under the section there might be a little arrow in the lens system that points to a cell or an area of the slide. They have to name that cell, name the function of that cell, or name a specific component arrowed in the section. And that's how histology was examined up until a few years ago. And one of the problems with that is that, firstly, it takes an enormous amount of time to set up. Secondly, when you've got 40 students moving from one station to another, they invariably move the slide or knock the slide. And so what was arrowed, what item was arrowed for students to identify, has been moved. So you've got to reset it, and it causes lots of dilemma. The other problem is, when you teach histology, traditionally, in what I call the old way, students don't actually have all the slides in their slide box that depict all the characteristics of organs. We have some very special slides that we only put out each week, depending on the topic, that are demonstration slides. And they will never see those slides again, particularly as they get towards the exam time, because it just takes too much effort to reset them up for demonstrations at the back of the class. It's too labour intensive. So that's how the exam was originally provided. Now, with 370 students, there's no way in the world you could have an exam where you're having 40 students rotating through 40 microscopes times 2 minutes to cover 370 students. There'd be hours and hours you'd have to allocate in an exam timetable. What do we do now? Well, new technology has enabled new methods of assessment. Students are happier because they do better. They're happier because they can constantly, daily review their own progress through their histology course, because it's all online. Previously, it was just too much work to set up demonstrations and very extensive testing using traditional practical laboratory time. On the website, electronically, students can access a number of different assessment packages. First of all, here, I've used blood as an example. Students have an extensive number of questions of the multiple choice type question, which they click on the button as they do this exam. And this test, by the way, is one they can do whenever they like, when they finish the module on blood that they have completed. Usually they're not allowed to do the test until they have spent sufficient time on the website to ensure that they've actually done the hard work before doing the test. And this test can be not only multiple choice questions, but it can also have other types of questions in. For instance, they roll down options and click that as their answer. Or they might have to type in the correct answer and spell it properly, or nearly spell it properly. And students can do these quizzes at the end of each of their topics, and those quizzes are very extensive. They can be up to 100 questions. And I say to them, look, there is a limit to how many different times a question can be asked or in a different way. So if you do these quizzes to help your learning, then you should do very, very well in the final exam. I use this sort of assessment as part of their learning process. At the end of doing these quizzes, they can go down the bottom and submit their answers, and they get feedback as to what they got right and what they got wrong. They can do the test as many times as they like. And at the end of that test, when they do it over and over again, and by the way, the questions can be randomly chosen, so they're not necessarily being asked the same questions all the time. When they do these tests over and over again and the learning, they really do know their histology. There is another sort of a test they can do. And this test can be merely accessing images with labels on it, and they answer the questions and they compare the answers later on with an answer sheet that they can download as well. There is another package, and that is shown here. They can go down to the questions and they can choose, in the yellow frame you see here, they can choose a topic. If you look at skin, it's got a little button next to it. They can set themselves a question by clicking the Set a Question box. An image comes up with a question. They can write the answer in, and then they think, I think that's the answer. So they can click, I think that's it. They get an answer response. It says, sorry, you're not correct. It gives them the correct answer. And again, there are hundreds of these questions randomly selected when they select each topic that they want to test themselves with. And let me emphasize again, when students do all these 3 or 4 different ways of assessing their progress, again, they do very, very well in their final examinations. Going back to the image scope, which is the last area that I wanted to concentrate on because I want some time for questions. But when students access the digital or the virtual microscopy database, I can now involve them in a lot more assessment fun. For instance, this is a section of skin. I can get the students to access their slide of skin, and I can give them a list of items of histological structures like sweat glands or sebaceous glands or little arteries, little veins, anything that's characteristically in skin. I can give them a list of these structures. And for an assessment, I can say to them, I want you to submit to me an image of each of these structures. Rather than the old way of having an image of a structure pasted in an exam venue with a label on it and asking students to identify this structure, might be a little artery, why not get them to create the image themselves? It's fun. It's interactive, and they can even exchange images with their fellow students as part of their review for study interaction. So I can now give these students a list, and they can send me a list of their 10 images of little arteries, sebaceous glands, etc., etc. They've created themselves. And that could be their assessment or part of their assessment. I can go further. I can say, don't just send me a picture of an artery or a sweat gland or a sebaceous gland. I want you to label it. Label all the main components. And for each label, you get X marks. A lot more fun now in histology using these interactive and more innovative technologies that we can now provide for our students. Well, is it successful? Am I successful? Or are these technologies successful in changing the way in which we can, first of all, teach histology? And one of the prime reasons for me shifting into this direction was purely a cost-saving exercise, increased student numbers, but very costly resources that we could not afford to buy more of. So that's when I first went down this path of putting learning of histology online. I think I've been successful at doing that. Have the students been successful? I think they have as well. I think success is the path they're going rather than failure. And I admit that there are still plenty of new things that I can do, and I will be doing. This is just the start, really. Have a look at the next slide. I'm not worried about statistics at this stage, but this is a particular histology course I taught several years ago. I can't remember the numbers of students, but this shows you the profile of the results of the final histology course or final histology exam. This exam was presented traditionally, whereby the students came in over a 3-hour period, 30 or 40 students each hour. They sat down on the microscope, 40 microscopes. They had to identify tissues or organs down the microscope, or they had to identify items that were labelled or had points on them, or they had to write answers to short questions. These are the results. Some students did very well. The majority of students just passed, some failed. And I don't think it's necessarily because of the attitude of the students. I think it's partly because of the difficulty involved in teaching a successful histology course using just microscopes, just slides, and the inherent difficulty with providing them different assessment packages to help them review their progress through the course. Sure, you can put out each week you can put out unlabelled slides and get them to try and identify each slide, etc., but it's confined to their laboratory time, which is often very limiting for some students, particularly those who have to either have class clashes or jobs, etc. Last year, the results looked like this. A lot more students. One of the things that's happened in this course, in my histology course, is that the student numbers have gone from about 50 to 370, and that's far greater than any other corresponding unit. And I think it's because it's an online course, students have fun, they have this whole host of assessment packages that I showed you earlier, that as you see in the graph below, the success of the students has improved markedly. The exam pass rate, the exam average has improved. In fact, these results seem to conflict with other units in the faculty. I don't like to moderate the results. I think if students do the hard work by spending a lot of time going through all the assessment packages, and therefore they perform very well in exams because of that, then I think they deserve that credit. And also, I have been given feedback that when these students go out into pathology or into the clinics, they are very good at recognising organ and tissue structures. So it does work. just doesn't enable them to get very high exam results, but enables them, when they get into the workforce, to be successful at what the learning outcomes of a histology course are all about. My last point, I've said it before, is that learning histology has inherent difficulties. On the left-hand side, you see a tube. The tube's sectioned. This section has been cut in various orientations, and the slide showing that section has a number of profiles of different shapes on it, because what the students have to do is they have to interpret a three-dimensional structure from a two-dimensional image that you see on the right-hand side. This happens to be a section through a sweat gland in skin, and you can see the sweat gland has been cut, showing a number of profiles. A sweat gland is one coiled tube, just like the analogy I have down the bottom of a piece of spaghetti. When you cut through all that spaghetti, you're going to see profiles cut in different orientations, just like you do when you cut through sweat glands. Well, students do have difficulty with that. They have difficulty interpreting three-dimensional structure from two-dimensional images. And that leads me on to the next vision I have, and that is that I am going to move on to moving away, in fact, from looking at two-dimensional images, i.e. slides, histological sections, and trying to recreate three-dimensional reconstructions and animations to get around this inherent problem. And that's going to involve also some more exciting assessment packages that hopefully I can create. I'll finish there, and I'll thank you very much for your attention, and am happy to answer any questions. I'm happy to answer any email questions you have. But again, thank you to Matthew for letting me participate in this presentation, and I look forward to hearing from you. Thank you again.